go ahead and get started. Welcome to our webinar today. Thank you for joining us. We have a couple hundred people expected this afternoon, so we're very excited for this webinar and this presentation. I'm Gretchen Kemp, the Senior Director of Marketing here at GenomOncology, and I'll be serving as the moderator today for the webinar. At GenomOncology, we're passionate about the use of genomics to improve people's lives, and our collaboration with Genomenon helps us further that mission. I'm glad you could join us today to learn about how our partnership with Genomenon is revolutionizing the genomic interpretation and advancing precision medicine through comprehensive somatic and germline testing capabilities. Next slide, please. Before we begin, I want to encourage you to ask any and all questions via the Q&A panel that appears at the bottom of the Zoom webinar. You'll see it there on your bar at the bottom. Your questions always lead to great conversations for us, so we'll save about 10 to 15 minutes at the end of the presentation to go over those questions. If for some reason we don't get to your question, we will follow up with you offline for sure. Next slide. So without further delay, let's get started. I'd like to take just a minute or two to explain why this partnership between GenomOncology and Genomenon is so critical to advancing clinical genomics. Today, this collaboration represents a powerful synergy in precision medicine, one that is hard to successfully replicate in a way that is fully integrated and economically approachable. GenomOncology provides enterprise-grade reporting and automation workflows that streamline the entire clinical reporting process, while Genomenon brings a world-class knowledge base of the genetics literature, as well as expertly curated genes and variants. Together, we've created something unique, a unified platform that can largely automate NGS reporting. The GenomOncology Pathology Workbench software, along with the Genomenon knowledge base, supports reporting of both hereditary NGS assays, including gene panels, as well as whole exome, whole genome, and somatic NGS assays. This platform supports every aspect of NGS reporting. That includes quality control, variant annotation, variant classification and interpretation, and identification of therapies and other actionability, as well as fully customized reporting. And now it gives me great pleasure to introduce you to our webinar presenters today. Denice Belandres is Genomenon's, I'm having a real hard time today with GenomOncology and Genomenon, Genome's Senior Manager of Customer Success. She supports Mastermind CKB users across both clinical and research applications. With a background in germline variant interpretation in clinical NGS labs, she brings deep technical expertise and a passion for helping customers advance their work in both rare disease and oncology. Matt Stachowiak is the Vice President of Product Innovation at GenomOncology. He has over 10 years experience in precision oncology software and data products. Matt manages the innovation roadmap here and supervises initiatives in pathology, bioinformatics, therapy, clinical trial matching, and API design. Prior to GenomOncology, Matt obtained a PhD in chemical engineering and biophysics from Columbia University in New York. Welcome, Denice and Matt. Denice, I'll pass things over to you.
Awesome. Thanks so much, Gretchen. And thanks to all of you who have joined us for the webinar today. Really excited to talk to you all about Genomenon, our two platforms, CKB and Mastermind, and of course, our partnership with GenomOncology. You can go ahead and create your free accounts using the bit.ly links here that are displayed on the screen. Like Gretchen said, please drop your questions in the Q&A and we will address those toward the end of the webinar. Next slide, please. So Genomenon, we are a genomic intelligence company. We provide data and services for applications in both clinical diagnostics and precision medicine. Our mission is to make genomic information actionable, and we aim to simplify complex genetic data into actionable insights. Next slide, please. So we're doing this three main ways. That's through our software, data, and our services. So we're going to be focusing on Mastermind and CKB today, which are both software and they have their own UIs, but the data is also integrated seamlessly into GenomOncology's platform, which Matt is going to present later on as well. So we're using both contemporary AI tools and the genomic expertise of our people to deliver these offerings. That combination is really the foundation of what we do, and it's kind of a recurring theme, as you'll see on the next slide here, please. So our approach then is in leveraging our domain expertise, right? Our people alongside modern AI and sophisticated computational methods. So that's the combination, the approach that we believe really meets the moment, meets the challenge in clinical genomics right now, and it's what's setting our data apart. So in partnering with GenomOncology and integrating that data into their platform, we're really allowing care teams to understand all of the drivers of a patient's cancer and ensuring that no actionable genomic information is going to be overlooked. Next slide, please. We are really proud to work with organizations all over the world to help save and improve patient lives. We share this passion with GenomOncology, of course, and we're excited to share more about how our collaboration is advancing genomic interpretation and precision medicine. So we're going to start with an overview of Mastermind. You can hit the next slide. We'll walk through some overview details and walk through the application a bit before moving on to CKB. So we can move on to the next. So Mastermind is a genomic evidence platform that combines genomics-focused AI and expert human curation to support clinical decision-making and accelerate patient diagnosis. So the platform provides access to over 10 million full-text genomic articles and over 3 million supplemental data sets, and right now covering over 26 million genetic variants. In addition to that very comprehensive set of computationally gathered data, our team of scientists are also curating that evidence, so providing provisional classifications that are following ACMG guidelines. So then as a user, you get to access this short list of PMIDs, the ones that contain patients and families and functional data. So those very specific PMIDs that contain relevant information are often the first ones that you're going to be able to see. In the interface, you also get rapid insight into the literature with sentence fragments from the full text of these articles. So you'll be able to see your search terms highlighted and color-coded. You're also able to apply things like filters and additional search terms like phenotypes to get really, really specific results for your query. Next slide, please. So the three kind of main types of data or main components when it comes to Mastermind. And these data, it's available in our UI, it's available as flat files, and then of course the data are integrated into the GenomOncology solution. So there are curated gene-disease relationships and the literature supporting those GDR assessments. There's curated evidence at the variant level, and those curations are following ACMG guidelines. And then there's the indexed or computationally gathered information as well. So we have millions of articles in our database. Sometimes just knowing if there are any articles at all that mention your variant of interest, that's an important sort of first question to ask when you're beginning to investigate a variant. Next slide, please. So at a glance here, Mastermind is a highly comprehensive solution. We have extensive coverage for genes and variants, reach into millions of full-text articles and supplemental data sets, access to curated content like those gene-disease relationships. Matt is going to present more on how this data is integrated and available in the GenomOncology software. So I'll more on that when I pass the baton. But for now, next slide here. We're going to run through some screenshots of the Mastermind interface and show you how you can search across millions of articles to access really current information. We are updating Mastermind and adding new content to it every single week. We'll walk through how the curated ACMG evidence is displayed for variants, as well as the curated gene-disease relationships as well. So here I have searched for a variant in Rett as an example. So this is what it looks like when we've done that search. So front and center here is we have the curated variant evidence, and the summary of that curated evidence is circled in the middle. We'll zoom into that here in a second, but on the left here, I also wanted to highlight the number of articles that's retrieved for this search, which is 76. So sometimes just knowing that number is really helpful. The number of articles associated or available for a given variant. Is that number 0? Is it 500? That's a simple piece of information, but you can imagine a workflow where you might want to even automate the negative. So if the article count is 0, you might want to deprioritize and instead focus on variants that actually do have evidence or literature for you to review. So 76, that's quite a few articles. Lucky for us, this is a variant that has curated evidence, so that's where we'll start. On the next slide, please. So at the top of the screen here, the criteria kind of more broadly grouped clinical, functional, population, computational, and intrinsic. The ACMG criteria or codes are under each of these, so you can see what our curators have applied under each of those. And then we provide more detail below, specifically for clinical and functional evidence, because that's where we have literature that we have curated and additional information under each of those. So this is just, a screenshot, a snippet from the page. There's even more that we could scroll down and see, but I've circled just some of the discrete data that we've curated. For those PMIDs, just as an example, looking on the left column here, the first PMID under that clinical column, we're displaying here, there's one proband in this PMID. The variant is heterozygous in that individual, and the disease context is multiple endocrine neoplasia type 2A. So under that is another clinical study, but notice this one has in gray, it's listed as unweighted evidence. And in reading the reason that's provided there, we can see that there's a proband here, but there's no patient-specific phenotype information that was provided in that paper. So while this PMID is considered to be a clinical article, there's a patient in there, that patient has this variant. We haven't applied, or there's no ACMG criterion associated or tagged to that specific article. On the right side here, we're doing a similar thing for functional types of studies. So providing a sort of short description of the assay result, and of course, always providing users with link outs to PubMed, so you guys can take a deeper dive into those articles and review the literature. So curated data here, it's the most convenient way to interact with the data because it's highly specific. These PMIDs, they're categorized. We're not looking at a list of 76 PMID titles. We have a much shorter list of PMIDs here, and we have impressions of why each of them are important. So this is the kind of information you're also going to be able to see and leverage in the GenomOncology platform. But there's also a lot of indexed or computationally gathered evidence, including insights into the supplemental data that's available in the Mastermind UI. So that's where we're headed on the next slide here. So here in the UI, we're also going to flag articles that contain matches in the supplemental data with a paperclip. So that's what's circled there on the left side of the screen. So we're going to let you know that, hey, we found your gene or variant in the supplement of this paper. And then we're going to give you details, that's what's in the center here, about how that variant was described in that supplemental data, and then where you can go find it, all the way down to the name of the file where you can go and download it. Right above that is where you'll find the sentence fragments. So those are sentences or snippets from the full text. The variant is highlighted in blue. So you can get a peek at, how many times is the variant in the full text and get a little bit of context for how the author is describing that variant or how they're talking about it in that paper. And this is also where you could add additional search terms to your search, things like filters or add phenotype or even free text. The more things you add to the search, the more sentence fragments actually end up getting pulled out and displayed. In addition to variant information, which is what we've been focusing on so far, we also have gene level information in Mastermind. So in the top left where it says view ret details, if we click on that, we can go to the next slide and we will find ourselves on the gene page. So on this page, we have a variant diagram, and then we also have a variant table right below that. The diagram is interactive. You can click on those peaks. You can kind of zoom and scroll around. The table itself is searchable. It's sortable. And then there are icons in the table that indicate whether curated evidence is available for that variant, along with the number of articles that are available for that variant. So all of those, the little icons, the numbers, those are all links. So they're all sort of jumping off points to go and have a look at the curated evidence or see those indexed articles and those sentence fragments for those variants. Two more things on this page we'll call out that are on the next slide. So there's a section on the left here for a gene summary. That summary info can pop out if you click on that little rectangular icon. We'll look at it on the next slide. On the right, we also have gene-disease relationships. So these are the manually curated GDRs. So we'll take a look at both of those on the next slide, please. And this is the last slide before we jump into CKB, but under summary here, we have some basic information about the gene. Importantly here, we have some metrics for when you're using the ACMG gene criteria. So that's kind of aggregating data from lots of different sources if you're using those ACMG criteria. And then to the right, looking at the GDRs, these are those manually curated gene-disease relationships where we're displaying the name of the disease, the strength of that association, the inheritance pattern, and then all of the supporting PMIDs that are hyperlinked to take you to PubMed. So we're all about showing our work and bringing information to users as quickly as possible. That's sort of a theme. You'll see that with CKB as well. And so with that, we can go ahead and move on to the next slide. One more. So CKB. CKB is the standard in evidence-based interpretation of complex cancer genomic profiles, really enabling clinicians and researchers to search comprehensively across variants and treatments and trials, currently covering over 2,200 genes. The database is professionally curated and maintained by a team of PhD scientists, really to reflect the most current scientific evidence that's available. So it's structured for scalability. We're following standardized protocols, and really that's what's going to support reproducible interpretation. Next slide. So using CKB data, and Matt will show how this data has been integrated into the Genome Oncology platform, you can save time by accessing critical information quickly to identify and discover the best therapeutic options for your patients. With new oncology data emerging constantly, knowing that a team of experts is regularly updating and maintaining this curated information is really what's going to empower more confident clinical decision making. Next slide. In terms of the scope of CKB, so right now we have coverage in over 2,200 genes, 48,000 variants, 15,000 clinical trials, 4,000 therapies. We've curated over 43,000 unique efficacy evidence annotations. I'll be showing some screenshots of the CKB user interface here in a bit. And of course, Matt will show us how that data lives in the Geo workbench as well. Next slide. So we'll talk about how the data connects a molecular profile to a tumor type, therapy, and a response. We will show some of the visualization tools that are available in the UI for exploring variants by their impact and by the protein effect. And then, of course, showcase how we can support clinical decision-making by identifying therapies and clinical trials. So continuing on with RET here, this is the gene detail page in the CKB Boost application. The top left here, we have some high-level information, curated gene description. So that's going to include the gene's normal function and its role in cancer. You can see this is flagged as an oncogene. There are linked PMID references throughout. The bottom of this screen shows that there's a variant table. This is where you can find all the variants that we've curated to this gene with descriptions and also flags for association with drug resistance. A new feature in the UI is the variant navigator. We can kind of preview it on the right side, but we'll take a deeper look on the next slide. So this, we have on the x-axis amino acid position. There are also separate tracks below that for protein domains and regions and the exons as well. The y-axis here is number of variants at that position, and this is a lollipop graph. So when you're in the application, you can toggle between viewing those pie charts by impact or by protein effect. You can also zoom into specific regions. There's a variant table that lives right below this that's also going to provide that same variant information, right? The description of the variant also links to the variant detail pages. So actually don't have to navigate back and forth between this page and the gene page, so it makes it pretty easy to jump into variant info from here. Next slide, please. Another visualization tool in the Boost interface is what we call the sunburst plot. So that's related to a unique concept in CKB, which is a category variant. And a category variant is a group of variants that multiple variants can belong to. So if we're looking at a missense variant here, like this one, V804M, we might have, specific evidence for that specific variant, but there may also be extended evidence that we can look at the category level. So there's sort of two paths that we can follow to get to that extended evidence, and those paths are on the left side of the screen here. The top path is functional, so we go from the specific variant up one step to RET ACT mute, which is the bucket or the group of variants for gain of function variants in RET. The other path is positional, which is going from the specific missense to V804X, which is all missense variants at V804. And when we think about category variants, the way to visualize those is through the sunburst plot, which is on the right. The outermost layer there represents our individual variants. Our variant V804M lives in two places on that outer layer because there are two paths we can follow. But as we head toward the center, head toward RET there, we're encountering bigger slices. And those bigger slices are those category variants like RET ACT mute. And it's really this extended evidence or evidence that's at the category variant level that can provide a ton of additional actionable information when you're interpreting a molecular profile. So we're going to dig into that on the next slide here. So we have this tab for extended evidence, and this is where we're going to find a lot of that high-level evidence, things like FDA-approved therapies. So in this table is also where you're going to find those efficacy evidence annotations. So this is really the bread and butter of CKB. I mentioned earlier we have over 43,000 unique annotations, and this is where we're linking the molecular profile to a tumor type, therapy, and response. And it's really, distilling the evidence down to often a single sentence. So it's a manual curation, summarizing the data from the FDA label or the guideline or the clinical study. It's really how we're connecting the molecular profile to the therapy by the indication. Next slide, please. So this version of the sunburst plot, we're still looking at RET. It looks a bit different. So I've clicked on the ACT mute slice, and what that does is kind of re-renders the plot. So that's why it looks a bit different. So I can see now the member variants of that category variant. There's also a tab for viewing those member variants as well. But what I'm highlighting below is the associated clinical trials. And this is the last concept that I'll cover before handing it off to Matt. So in CKB, all the tables that you see are going to be filterable and sortable. So under clinical trials, there's actually 24 entries. But what I've done here is I've typed in medullary thyroid cancer. So that's acting like a filter right now. So that's leaving me with one clinical trial that fits that filter. Here on this page, the recruitment status is updated daily. And the data, everything that we're seeing here is coming from clinicaltrials.gov. There are more details that we've curated for this specific clinical trial. So when you click on the NCT ID in the left column, you're actually going to land on this other sort of detail page for that trial. You'll be able to see things like the info for the sponsor, the variant requirements, which could be an inclusion or exclusion criteria. And you'll be able to see the location of the trial sites as well. So lots of information, curated information for those clinical trials available in Boost. And so with that information and background on both CKB and Mastermind, thank you very much for your attention. I will hand it off to Matt.
All right, thank you, Denice. Hello, everybody. I'm Matt Stachowiak from GenomOncology. And so what I'm going to talk about is first, for those who are unfamiliar with GenomOncology, I'll do a very brief introduction to our company and our software. Then I'll give an overview of our partnership with Genomenon. And finally, I'll describe our tertiary analysis and reporting platform, which is called Pathology Workbench. And I'll show how the Genomenon data is a key part of this system. So GenomOncology is a software company. We're based in Cleveland, Ohio, and we've been in business for more than 13 years. We have clients throughout the United States and Europe. Many of them are laboratories, but also cancer centers, pharma companies, cancer-related charities, industry partners, etc. So we have a wide variety of software products available. Today I'm going to focus on our tertiary analysis software, so that's Go Pathology Workbench. But then note that all of the capabilities that I'm going to describe are also available via API access. So if you have built your own user interface, for example, and you want to just be able to make queries to get to get data, that's possible as well. And then just very briefly, we also have other types of software. We have what we call Ignite IQ, which takes unstructured data such as old PDF reports and automatically converts that into structured data. We have software to support molecular tumor board meetings. We have software called Precision Decision for oncologist decision support, and also for cancer centers to more efficiently manage their clinical trial evaluation and recruitment, as well as an analytics tool. We work in the AI space as well, et cetera. So many of our clients use multiple of our software products, both to help with the laboratory reporting, but also to help their organization as a whole, so that multiple different groups can easily work together to advance their precision medicine program. Okay, but now I'll narrow in on the tertiary analysis software. And so the idea behind Go Pathology Workbench is simple. It's taking as input first the raw sequencing data from an NGS assay, typically VCF files or the equivalent, plus any files containing QC metrics. And then also, secondly, patient and case information from the LIMS or EHR system, like the tumor type, if it's a somatic case, the phenotypes, the test ordered, the patient name, DOB, et cetera. And then once all that data is loaded into Go Pathology Workbench, that's the dotted box in the middle, there's 3 high-level steps. So the first step is QC review, where you can review all the QC metrics related to the run and to each sample on the run for positive control samples, et cetera. Then secondly, tertiary analysis, where for all the samples that passed QC, the software will automatically annotate each variant, interpret each variant, prioritize the variants for hereditary cases, identify actionability for the case, for somatic cases, meaning therapies, clinical trials, prognostic associations, diagnostic associations, and then finally create a draft report. And this tertiary analysis step is powered by the combination of our own knowledge base, along with the data from Genomenon CKB and Genomenon Mastermind that Denice just told us about. And then members of the lab can log into the software and review everything in their cases, make any tweaks if needed. And then finally, they sign out the report, at which point the report is delivered automatically to its destination, which is usually either a LIMS system or an EHR system via HL7 messaging, or both. And then finally, we also offer services when needed. In particular, we have an interpretation services offering where our team can do the initial review of each case in the Pathology Workbench software, so that when the pathologist from the lab logs in, the first thing that they would see is a sign-out ready report. Okay, so onto our partnership with Genomenon. So if you're a lab doing clinical reporting, then in addition to the knowledge base, you also need a software system to manage the data, a database of your final classifications for variants, to manage your case workflow, to keep an audit trail, integrate with the EHR, generate a customized report. And this is what Pathology Workbench does. So the idea behind our collaboration with Genomenon is that if you license the Genome-Oncology Pathology Workbench, the Genomenon data can be included. And it's built directly into the software so that both the complete knowledge base and the case reporting workflow are all handled in a single system. And this has many benefits, the first of which is that it's more cost efficient to do it this way compared to licensing various things separately. It's also time efficient in the sense that we do all the work of loading the Genomenon data into our software, automatically surfacing it for a given case when appropriate using our matching logic, and then automatically including it on the report in a sign-out ready way. And then note that the Genomenon data is included both for somatic testing, so that's the Genomenon CKB, and also for hereditary testing, Genomenon Mastermind, as Denice showed us. Okay, so now I will quickly go through some of the core features of Pathology Workbench itself, and then I'll finish off by showing more details on how the Genomenon data is included. So first, Pathology Workbench is assay agnostic. It can support any assay, whether it's commercially available or a custom assay, whether it's a somatic or a germline assay. And the software was built from the beginning to be easily extensible. So it's not a large lift in terms of time and expense for us to support any new assay that comes out, or to support any custom assay that a lab may be doing. And we will also use whatever data files are generated by a given assay. So we'll never ask the lab to transform VCF files or other data files to a more convenient format for us. We can very rapidly, assimilate to any type of output. All right, next, in addition to SMVs and indels, the software supports all types of biomarkers. So everything you can do by NGS, copy number alterations, fusions, splice variants, all of the molecular signatures, pharmacogenomics, etc. But then also non-NGS results can be included. So for example, several labs do PD-L1 IHC for certain solid tumor cases, and they include those on the same report along with the NGS results. Or some labs do a FISH panel, and include that on the same report as a heme NGS panel. And these non-NGS results, PD-L1 expression, fusions by FISH, these are fully taken into account in the case interpretation. So they would match to therapies, clinical trials. They'd be taken into account for prognostic associations, diagnostic associations, etc. Okay, so now the knowledge base. So the reporting is powered by the comprehensive knowledge base. And of course, the key thing here is the incorporation of the Genomenon data for both somatic and germline purposes. But then also in the software, each variant is also annotated with many other data sources to provide a complete picture. So for example, the software calculates proper HGVS nomenclature. ClinVar is built in, ClinGen, Genie, population frequencies, and many other data sources. And so all of this together is everything that you need in order to automatically generate an accurate and complete clinical report. And then the report itself is fully custom. So as a standard part of implementation, we will customize the report for your lab. And as you can see by the examples on the screen, it's not just taking your logo and putting it onto our default report. Really anything is possible. You can also have multiple report templates, a different report for somatic versus germline, or a different report template for different assays. Any number of report templates is possible. And you can also do multiple report formats. So a PDF report, or an XML or a text file, or for those labs unfortunate enough that they still have to copy paste into an EHR or a LIMS, Microsoft Word, and we can tailor the report to make it very easy to do the copy pasting until we're able to do the full integration. So lots of possibilities there. And then Pathology Workbench has been live for over 10 years. And so over time, it's been continuously improving so that it supports, the whole range of scenarios that you encounter in the lab. So we have a ton of experience integrating with Epic for automated ordering and then the automatic returning of results back to Epic via HL7 messaging. And we build those HL7 messages to your specification to make it very easy for your Epic team and your IT team. We will do whatever they need to minimize their effort and involvement. And we're actually planning a webinar for October where we will go into great detail on this subject of integration with the EHR. So if you're interested, keep an eye out for that. The Workbench also has an API that can be used to automate integration with LIMS or list systems when that makes sense. The software handles, as I mentioned, QC metric tracking and review. It has IGV built in for BAM file inspection. And then it handles various reporting scenarios, such as a single analyte report. So if you, just want to report out your fish panel first, in its own report, and then later also include it in the integrated report, we can do that. It handles addendum and amendment reports. It handles QNS reports, if the quality or quantity is not sufficient. And then less common scenarios as well, like reflex testing or tumor normal pair. So you can really manage your whole process in the software. It's not only annotation and reporting, but also your whole workflow is in the same spot. All right, and then finally, the actual hosting of the software is also completely flexible. So the software can be installed on-prem behind your firewall, on a server that you provide, or it could be installed on a cloud server that you provide, or it could be installed on a cloud server that we provide if you prefer us to do that. And regardless of which option is chosen, you get a dedicated server just for your lab. So the only data that's on that server is the data from your lab. And so this is what one of the things that enables our flexible approach to customizations, like the reports, and there's a lot of tweaking that can be done in the configuration to set up the workflow just the way you like it, and that's because the install of the software is just for your lab. And this also enables us to schedule software and database updates according to the lab's preference. So we know that major software updates can create validation headaches, right? You have to revalidate in certain scenarios. And so we don't force our clients to accept software updates. Software updates happen only with the lab's approval on their schedule, to eliminate all those validation headaches. And then, finally, your data remains yours. We don't take data rights, and your data is not in any way locked in our software. All of the data is easily accessible and exportable from the software at any time. Okay, so moving on to a bit more detail about what the integration with Genomenon actually looks like in the software, starting with the hereditary side. So first, what's included? So that's all of the ACMG variant classifications done by Genomenon's expert curators, and that's currently at more than 1,200 genes. It also includes all of the manually curated gene disease relationships, of which there are currently more than 9,000. And it also includes link outs directly from GenomOncology to the Mastermind user interface for when a deeper investigation of the literature is required. And so this first screenshot shows a variant from the main variant grid within Pathology Workbench for a hereditary cancer case. And this is a missense variant in Rett. It's the same variant Denice showed earlier. And we can see that this has been classified as pathogenic by Genomenon. And this is extremely helpful because this particular variant, is not in ClinGen. It's not, and it's considered VUS by by the submitters to ClinVar. And so, Genomenon's gone and done the work of the deeper investigation needed. And then we can also see in the GDR column, there's gene disease relationships from Genomenon, which include the multiple endocrine neoplasia associations, which are in ClinGen, but Genomenon has many additional GDRs that could be helpful. In this case, the Hirschsprung disease. Yeah. Okay, and then the next screenshots over here on the right are from the variant detail window. And here we see the pathogenic classification, as well as the count of papers in Mastermind, along with a link out to Mastermind, as I mentioned, to directly view these articles in the Mastermind user interface. And then if you click on this pathogenic link here, we've also incorporated directly into the software all of the details of the Genomenon classification that Denice showed earlier. So for each variant classification, here we see it's pathogenic, but we also see all of the ACMG codes that were applied and specifically which references were used. So in this case, multiple codes were applied, which altogether result in the pathogenic classification. Okay, so now I'll switch over to the somatic side, and the primary things included here would be the protein effects, meaning Genomenon's curation of the literature as to whether a given variant is gain or loss of function or has no effect. And currently there's more than 48,000 of those in the database. Additionally, gene descriptions and mutation descriptions, which are very helpful to the report, as we'll see. Resistance therapies, efficacy evidence, drug descriptions, and then similar to the hereditary side, a link out to the Mastermind site if deeper investigation is needed. And then finally, using this Genomenon data on the somatic side, as well as our knowledge base with other sources, our system, the pathology workbench, automatically classifies each variant for oncogenicity. So is the variant oncogenic, likely oncogenic, uncertain, likely benign or benign? And it also automatically calculates the AMP ASCO cap tiers, which relate to actionability. And so one of the larger labs that we work with has done a concordance study on this automated classification. And, we're at 99% concordance with manual classification that their variant scientists have done over the previous several years. And you can learn more about this study at the upcoming AMP and ASH meetings in November. There'll be posters and talks about that. Okay, so moving on, here's the main variant grid for a somatic case. And we see another RET mutation, V804M. And this has been called a gain of function by Genomenon. And when we open the detail window here, we see the details of this gain of function call, including the PMIDs, and then similar, a link out to the Mastermind UI as well, just as we had for the hereditary case. And then here on the PDF report itself, we can see, and again, the report is fully customizable, but our default template includes the protein effect from Genomenon, and then automatically includes the Genomenon gene and variant descriptions, so that composing narrative text relating to each gene and variant is not a task that the lab has to undertake. This all happens automatically. Although, of course, in the software, you're able to write your own text if you want to, but the Genomenon is preloaded for this purpose. Okay, and that concludes my section. So Gretchen, I will pass it back to you.
Great, thanks, Matt. You both did excellent presentations. I appreciate that. I hope you all gained some valuable learnings from these presentations today. This partnership is particularly important because modern precision medicine requires seamless integration of comprehensive somatic tumor profiling, along with germline risk assessments for follow-up recommendations. That partnership also needs to be done in a way that provides premium quality data that is economically accessible to all providers and patients. Patients benefit when their care teams, via the appropriate testing, can simultaneously understand the genomic drivers of their current cancer, while also identifying hereditary factors that may impact things like treatment decisions, family counseling, and long-term surveillance strategies. Our combined platforms enable this comprehensive approach, ensuring that no actionable genomic information is overlooked. If you take nothing else away today, I hope you walk away with the knowledge of all of that after today's webinar. It's now time for our Q&A. Next slide, please. As a reminder, ask any questions that you have in the Q&A dialog box down there on the bar in the Zoom panel. It says Q&A. We'll get to as many of these as we can. And what we don't get to, we'll follow up directly with you. We do have your information on the side and can do that. So looks like we have a couple questions already coming in. So I'm going to go ahead and start to share the first one. And this one goes to Denice. Who does the curation at Genomenon?
Yeah, so as a whole, Genomenon's curation team is over 100 master's and PhD level scientists. They're full-time curators. There's sort of two main teams, as you can imagine, to support the Mastermind curation and the CKB curation. But within those teams, we also have kind of more specialized folks and QA leads. Of course, there's multiple rounds of review before we actually end up pushing data into our platforms. But if there are, specific questions about the team and their background, would love to set up a chat with whoever asked the question. I mentioned too that we have services that we offer at Genomenon, and our curation team is really the star of those engagements, obviously. So always happy to introduce our folks to whoever wants to meet them. That's kind of the great thing about having manual curation, is you can talk to the people who did the work. So yeah, we'll definitely welcome the feedback on our curated content through either our support emails or the buttons in our UI soliciting your feedback. But yeah, we have a pretty large team doing that curation work full time.
Awesome. Thank you. This next question is actually to both of you. How often is the data updated? Denise, I'll ask it to you first, and then Matt, if you want to answer as well.
Yeah, I'll speak to the UI piece and then Matt to the integration piece. So for Mastermind, the data is updated every week. So new articles get added to Mastermind every week. New curated content is getting added usually every week. In terms of maintenance of things that we've already curated, those are typically reviewed yearly. So that's on the Mastermind side. On the CKB side, new content in Boost is updated every day, including those clinical trial recruitment status. So that's daily. For CKB, we'll review guidelines every month, and we'll make content updates if there are any changes to those guidelines. And then for variants of uncertain significance in Boost, we're going to check for new literature every year. So it's kind of multi-part question for Mastermind and CKB, but I'll let Matt speak to the integration piece.
Yeah, and then within Genome Oncology, it's typically a monthly release. So on a monthly basis, we'll take the latest version of the Genomenon data and incorporate that into our software.
This next question is for Denise. Are the articles listed already have full access?
Yeah, I believe this was asked during the curated content section of Mastermind. So I'll answer from a couple angles because I'm not positive I understand correctly. But so if the question is whether the articles that we curate are always open access, not necessarily. So our team does have access to articles that are behind a paywall. So we have access to them, we're curating them, and then we're passing on that curated information to you in the UI and through the integration. So you're able to see paywalled articles. We cannot then in turn, provide you the PDF of a paywalled article just because we have access to it. So we can't do that. But when we provide those links in Mastermind, when you see a PMID link in our UI and you click on it, that takes you to PubMed. So that article may or may not be open access. Your ability to access the PDF depends on whether you have a subscription. But the other aspect to answering this question is the sentence fragments. So I showed those. So let's say that you don't have access to a paper. Those sentence fragments that you can see in the Mastermind interface, that will tell you how many times your variant is in there. It gives you some of the context around how the variants being talked about. And if you add more stuff to your search, like a disease and keywords and filters and things like that, you end up pulling more of these sentences. So the sentence fragments, of course, they're not going to be a replacement for getting the full text. But the hope is that it could save you some time and save you from potentially buying a paper that would otherwise be a waste of time. So, if you are going to spend the time and the money to go get a paper that's behind a paywall, hopefully the sentence fragments will have given you that assurance that, hey, that paper talks about my variant in a context that I care about.
It's kind of like the concept of try before you buy, right?
Yeah, exactly.
Matt, this one's for you. In terms of concordance, is it for CAP, AMP, ASCO classifications? If yes, how was your experience with classifying tier 2 variants?
Yeah, the concordance study was focused mainly on oncogenicity. Is the variant oncogenic? Biologically speaking, sort of intrinsic to the variant. Is it oncogenic, VUS, or benign, likely benign? The AMP ASCO CAP tiers, which focus more on actionability. Is there a therapy? Is there a trial? Is there prognostic significance? Those, by their nature, are going to be less concordant across different labs. There's all sorts of questions with that system. If there's yeah, so there's a lot more judgment calls. So this study was focused on oncogenicity. But yeah, tier two variants is always a discussion point with labs. Is how do you want to handle them, and also do you consider what do you do with a variant that's oncogenic, but that doesn't have really established therapeutic, prognostic, or diagnostic significance. And different labs have different approaches. Some labs, in their mind, if it's oncogenic, it's tier two. It doesn't matter if there's actually the therapeutic, prognostic, or diagnostic. Other labs are comfortable with saying, okay, it's oncogenic, but yet it's still tier three because there is no known impact. And anyway, my main point is that our because of the report is customizable, our software is flexible, and we can make it work and treat tier two variants and tier three variants in whatever your desire is. But yeah, the concordance study was focused on oncogenicity, and we'll be able to talk about that in a lot more detail at or after AMP and ASH in November. Thanks for the question.
Yeah. Don't go too far. I have one more question for you. Can you comment on how comprehensively genes related to hematologic and lymphoid neoplasms are covered in your Genomenon database?
Yeah, so I'll answer that first, even though it's for Genomenon.
Sorry.
I can say that some of our largest volume, some of the largest volume labs using our software are focused only on heme at very high volumes, and so yes, heme is fully supported, as is liquid biopsy, as is solid tumor. So yes, for somatic, any type of somatic test can be fully supported by this system.
And Denice, do you have anything to add to that?
Nope, not much more to add to that. Fully agree.
Denice, this one's for you. Do you get CKB boost in Go?
So the boost data, as Matt showed, is fully integrated, right? So the exception would be the visuals that I showed from the boost interface. So while you don't automatically get a separate boost subscription, that's definitely something that you can talk to the Genomenon team about, and we can work to get access to that to the boost interface for you, for those who are interested in getting access to those visuals and other features that are unique to the interface. But other than that, the data itself is in Go, so it's just really the visuals that are separate, that would require a separate subscription to the boost.
Great. Well, we have a couple more questions, but we are getting to the top of the hour here. So for those of you who have asked a couple questions, we will get back to you through the emails that you've provided during registration. I'd like to thank all of you today for taking the time to participate in our webinar. Thanks to Denice and Matt for their very insightful and informative presentations. This webinar will be housed on both of our websites for you to be able to go back and listen again. So we encourage you to do that. Always good to reinforce information that you hear sometimes second and third time. So we invite you to go and do that. I wish you a good rest of your day, and thank you so much. Take care.